No, it is. Yes.
It says broadcast is live.
Yes.
Okay. Let it be visible on the page. 30 सेकंड का गैप आएगा.
अच्छा अच्छा अच्छा.
वैसे लाइव तो हो गया होया.
हाँ हाँ.
Okay. We can start.
Yeah.
Prof, we can start.
No, no. You have to start, no?
Okay, so today I welcome all of you in the once again in the Association of Microbiologists of India-Chandigarh weekend lecture series. I have few before we start today's lecture. I have few announcements to make. First of all, I am thankful to all for giving immense support as we have successfully completed the 1 month of this lecture series. During this 1 month, we conducted 9 webinars. One was the inaugural webinars, and 8 webinars were conducted on Saturday lecture series and Sunday journal club series, in which various eminent scientists and young faculty gave their lectures. So I'm thankful to all. I'm thankful to the faculty department of microbiology. I'm thankful to all the team members of AMI Chandigarh unit. I'm thankful to all the speakers. And I'm thankful to all who joined live during this session, the students, faculty, and all. Now, as the teaching has started, we have merged the Saturday Sunday lecture series into Sunday only. So we will be having lecture every Sunday from 12 to 1. Some of the lectures will be given by the senior scientists. In some of the slots, we will be giving opportunity to the young faculty. So in this new phase of our weekend lecture series, today we are having the lecture of Professor Rajat. Now I over the program to Professor S.K. Soni to welcome today's speaker and all those who have joined live, and also to introduce today's speaker. Professor Soni, please.
Thank you, Dr. Naveen Gupta. So, good afternoon to all of you on the 9th lecture of the weekly lecture series launched jointly by AMI Chandigarh Unit and Department of Microbiology, Panjab University, Chandigarh. And we have with us Dr. Rajat Sandhir, the speaker for today's program. Dr. Prince Sharma, Professor, Department of Microbiology, PU, Dean, Faculty of Science, Panjab University, and President of Chandigarh Unit of AMI. That Kusam Azhari, Chairperson, Department of Microbiology, Panjab University. That Naveen Gupta from Department of Microbiology, Panjab University, who is also the General Secretary of Chandigarh Unit of AMI. The members of AMI and other participants who have joined live on Facebook. I welcome all of you on behalf of the Chandigarh Unit of AMI and the Department of Microbiology. So let's start the today's proceedings with the introduction of the speaker, who is Professor in the Department of Biochemistry, Panjab University, Chandigarh, and also the sitting member of Panjab University Senate. Professor Rajat Sandhir, besides obtaining his master's and PhD degrees in biochemistry from Postgraduate Institute of Medical Education and Research, that is PGI, Chandigarh, obtained for postdoctoral experience from Medical University of South Carolina, US, and Kansas State Medical Center, USA. His areas of interest and expertise include neurobiology of disease and neuroprotection. So, Dr. Sandhir has more than 25 years of teaching and research experience. He has guided 25 PhD students from India and abroad for their research work and handled various research projects funded by different national agencies like ICR, DBT, DHC, UGC, etc. He has published more than 150 research papers in international journals of high impact factors with citations of more than 3,000 and H-index of 31. He is a fellow of Indian Academy of Neuroscience and Indian Association of Biomedical Scientists. He has served as editor Panjab University Research Journal in Science, member editorial board, World Journal of Diabetes and Toxicology International, executive member of the Indian Academy of Neurosciences. He is a member of various professional bodies including International Brain Research Organization, Indian Academy of Neurosciences, Society for Neurochemistry India, Society of Biological Chemists, Indian Biophysical Society, Society for Mitochondrial Research and Medicine India, He has successfully held various administrative positions in the past, including Chairperson, Department of Biochemistry, Coordinator, Institute of Forensic Science and Criminology, Coordinator, Center for Nuclear Medicine, Head and Coordinator, Central Animal House of Panjab University, Chandigarh. He is a Fellow, Panjab University Senate, Member, Research Promotion Cell, Panjab University, Chandigarh, Coordinator, UGC SAP for DRS Phase 1 and 2 in the Department of Biochemistry, Punjab University, Chandigarh. Program Coordinator, DSC 5th in the Department of Biochemistry, Punjab University, Chandigarh. Besides a good teacher and researcher, he is also a good human being. Dr. Sandhir will be talking about a very important topic of the present day scenario when the world is facing the problem of COVID-19 pandemic. I now invite Professor Sandhir to start his lecture on an overview of COVID-19 pathogenesis and treatment. So, Dr. Rajat, please.
Very good afternoon to you all. I would make a disclosure in the very beginning. See, COVID-19, world is struggling to know about it. Even I am trying to understand about COVID-19. It's not area of my interest, but I'm shifting my interest towards COVID-19. So that's the disclosure I would like to make. And if I fumble, there could be two reasons. Speaking among your peers is always difficult, and you will be a little more conscious. And then I said, I'm not expert from COVID. I'm learning COVID along with you people. Even WHO has been struggling to understand about COVID. And about AMI and microbiology, microbiology department has been amongst the forefront departments of Punjab University in terms of their output, in terms of quality of students they produce. And I know AMI Chandigarh unit has been performing excellent, and they've been ranked for probably 5 consecutive years as #1 chapter in the country. And that speaks volume about novelty in ways they bring out new programs. And I'm impressed with the Sunday seminar series that they have initiated. And with these words, I would get started with my presentation with you. And I'm going to give you an overview. My lecture will probably not be complete, but you can say that I would sensitize you on some aspect, which may be something new about COVID-19 or coronavirus that we talk of now. And I'll take a minute to share my slides. Are my slides visible?
Yes.
Okay.
Dr. Rajat?
So I put my title as Overview of COVID-19 Pathogenesis and Treatment. And I will get started with this particular slide. Many of you are familiar with this. The name coronavirus comes from the protein this virus has on its surface called spike proteins, and they give it a crown-like appearance, and that's the reason it's named as coronavirus. It's a Latin word. Corona means crown. And if you go back to the history of coronaviruses, there have been coronaviruses that have been affecting humans. And I would like to cite 2 examples of this. One was SARS, severe acute respiratory syndrome, that came in 2002, lasted for almost a year, impacted maybe thousands of lives. Then there was Middle East respiratory syndrome that came in 2012, and it originated from Middle East. That's why the name of this virus is MERS. And it went to, I would say, Korea, and this also did not have that much of mortality. And this kind of epidemic also lasted for a short time. Now we are facing another challenge in humanity in the form of this coronavirus leading to a disease called COVID-19, which the name comes from novel coronavirus disease. And the virus is called as SARS-CoV-2. And this is a new coronavirus which emerged from Hubei province of China in December 2019. And there is, sorry. So there's no part of human life that has not been impacted by this particular virus. And this is one of the transmission electron microscopy image of SARS-CoV-2 virus. Some of you might have seen this picture. I just want to show you one thing in this picture is these spike-like or crown-like projections on the surface of this virus, which is characteristic of these coronaviruses. If you look at this virus, it is 120 nanometer long. RNA is the genetic material, which almost weighs 0.85 attograms, which is 10 raised power minus 18 gram. And you can see this smallest entity has brought humankind to its knees. So I will like to quote one of the dohas, which one of my teachers said long back for microbiology, dekhan mein chota lage, gam de gambhir. Or you can say microbes or viruses can lead to devastating effects. And if you see, one individual could be infected by 70 billion viruses, and the weight of this would be 0.006 zeros and 5 grams, and would make one person sick. 2 grams are enough to infect 4 million humans. The world has been turned upside down by this small organism. And you all are probably aware of this, that the common symptoms that we can see from coronavirus is fever, cough, sore throat, shortness of breath. These are classical symptoms, or you could say some of the symptoms that we mostly talk about. And then we know there can be severe conditions it can lead to. And without much ado about the symptoms and things, this is the beautifully looking virus. But I want to highlight on this few things in this virus is it contains 29,000 plus nucleotides, which codes for 29 different proteins. And majorly, these are the structural proteins in this virus are spike proteins, the envelope protein, the membrane protein, and nucleocapsid protein, which is enclosed along with the viral RNA. And if you look at this genome, translates into the structural proteins shown in this yellow color, S, E, M, and N. These are all structural proteins. Then there are certain proteins called accessory proteins. And then there are non-structural proteins named as NSPs, NSP2, 3, 4. I will like to highlight about some of the proteins this virus would have. And about the structural proteins, these are the membrane protein, the spike proteins, the envelope proteins, and then there is an hemagglutination esterase. These are called as structural proteins of the virus. So one of the most interesting protein of this virus, which is even giving it the name of the virus, is the spike protein, which is a glycoprotein, and it forms the crown of the virus. And this is what is responsible for entry of this virus into the host cell. As protein, interestingly, is cleaved by a protease from the host, which is abbreviated as TMP-RSS2, which is a furin-like protease, and which cleaves this spike protein into two domains, S1 and S2. This S1 and S2 cleavage is very, very critical in facilitating the entry of this virus into the host. And S1 is known to attach to the cellular receptor, which is ACE2. ACE2 is angiotensin converting enzyme 2. I'll talk about this a little later. And then S2 leads to fusion of the viral and cellular membranes. That means it is the spike proteins that would be very, very important in interaction of this virus, virus with the host machinery. Then there are membrane proteins, most abundant protein, and interestingly, this spans the membrane 3 times and has an extracellular end region and intracellular carboxyl domain. And its primary function is to generate virus-specific humoral response, or you can say immune response for people who are going to be laymen's. And this is the most abundant structural protein, and it neutralizes antibodies developed in the SARS patients. So that means with this protein, the virus is going to be able to survive because it can neutralize the antibodies that are developed in these patients. And the transmembrane domains transmembrane domain is these 3 loops, contains a T cell epitope cluster that holds the cellular immunogenicity against from the host cells. Then there are envelope proteins, and these are transmembrane again. These form channels allowing human protease to release the viral genome material to the host cell. So this is again facilitating the release of genomic material to the host cell, and this is not important for viral replication, but important for pathogenesis, because it helps in assembly and release of the virus. So you can see all these 3 or 4 proteins that I talked are important in the invasion of the or entry of the virus into the host cell. Then there are nucleocapsid proteins. I said these would be part of the RNA complex, and these also give bead on string kind of conformation, almost something similar that you see with the histone proteins in the eukaryotic cell. And this protein is phosphorylated, and it encapsulates the genomic material in the virus, and it ultimately forms a complex with replicase complex, which is important for transcription or copying of the RNA into the virus or assembly of the virus. Then another protein called heme agglutinase esterase, which is a dimer. It contains esterase activity. This is the protein that would help in binding of sialic acid residues on the surface of host proteins and assist in release of virus from the infected cells. Then I talked about non-structural proteins. I'll talk about 2 non-structural proteins that have been very, very critical. One of them is called main protease. It's abbreviated as NSP5. If you go back to the slide I showed, where I showed the genome and the proteins coming from this genome, I said there are structural proteins, and then there are non-structural proteins, and there are many non-structural proteins, and I will talk about 2 major of these which have a critical role in this. It's called main proteins, abbreviated as MPRO, also called as 3CL-Pro, and which, along with a papain-like protease, is essential for processing of polyproteins that are translated from the viral genome. So that means this is also a protein that is critical to virus multiplication. Then we have this most critical RNA-dependent RNA polymerase coming from the protein abbreviated as NSP12. And interestingly, I said this is a RNA-dependent RNA polymerase. This protein itself has minimal activity, but when it, along with NSP7 and 8, its activity gets enhanced. So that means for full activity to be there, you need a complex of NSP12, NSP7, NSP8. And then talking about the strains of this virus, if you follow the literature, it was said that there are 2 major kinds of strain this coronavirus have. One is called S, another is called L. 70% of the patients were said to have the L strain, and L strain was more lethal, and it was the one which was associated with majority of the patients that were seen in Wuhan. And this was the other 30% of the patients had this S strain, which is less aggressive, slow spreading, and causes milder illness. And you can see the mutations in this. There is a cytosine that has been replaced by thymine at position in the spike protein, and this is another one. In L strain, we expect change at 28,144 base pair from T to C. Change is what is seen. Interestingly, this S and L nomenclature has been now kind of replacing with a classification of viruses, where we're trying to classify them as A, B, C, because of the way people had classified their different kinds of nomenclature that came up. So then it was considered that we should have a uniform system of classifying these viruses. So it was classified as A, B, C. A is the closest to, the virus is closest to that is seen in bats and pangolins, from which these coronavirus appeared. And this is the virus. A type is the one which is the root cause of the outbreak. And then type B is the variation from type A, and it mutates slowly, but it has been seen that it mutated rapidly outside China. Type C is daughter of B, and there is a variation that is there in this, and this is what is then another strain that is circulating in the world now. If you look at the global map, you can see all A, B, C type of these coronavirus strains are there in different parts of the world. And further than this, there are further mutations to these strains, and we've seen that there are further they are subclassified into different subtypes of these viruses. This is a diversity map of SARS-CoV-2 from India. You can see in different states we have different mutations in this virus. Some of them are B1 type, B24 type, A1, A3-1 type, A2A type, A1A, and A3 types. So that means we are seeing so many variations of this virus within India itself. And you can say probably there could be thousands of different mutations you can expect in this virus. This is a phylogenetic network of SARS-2 genome. This is where the bat is, black dot at the bottom of this slide. And you can see within the world now how many different strains of this virus we are encountering. So the message here is that when you have so much of variation happening in the virus, there will be a big challenge in developing vaccine against these coronavirus also. You can see, if you go back to the timelines of this, December 31, Wuhan reported 27 unknown pneumonia cases. This is talking about the history and how this virus spread. And some had close contact with human seafood wholesale market. We probably have been following news. We know this. Some reports were there in December 2019, and you can see how the numbers started growing. And this is the data from till February 2020, and we know by this time it was almost spreading to the world. And March, this virus was declared as pandemic, and this is the global spread of this virus now, with 23 million cases worldwide and almost getting to 1 million death. And I said 23 million worldwide, and you can see India is also having now 3 million cases, and the number of casualties have been more than 56,000 now. And India's COVID tally in August is highest in the world so far. We've added 10 lakh cases in last 10 to 15 days. So this is the graph which is trying to tell that the COVID-19 infections are rising steeply in India. I want to particularly draw your attention to this graph, the green line. The green line is representing the infection in South Korea. Look at how this green line almost dipped down within a couple of months. This red line is for India, and this data is almost 1.5 2 months old data, and you can expect this red line is even increasing steeply. And these are the projections that has been said for India. India might have 2.87 lakh COVID cases per day by February 2021. And I believe we are all witnessing this, and probably will, whatever is projected, this was projected 2 months back, we are almost inching towards these numbers. Seems like within months, we might have 1 lakh cases a day. Anyway, the whole idea about this COVID has been to flatten the curve. When you flatten the curve, probably expecting that you can prepare things better. This is normally you expect if you don't have the lockdown, you had the graph like this. But if you had the lockdown or have restrictions, you can reduce the number of cases. But the number of cases might remain the same, but you can flatten the curve over a period of longer period of time. That's what is flattening the curve means. We need to understand that it is not the number of cases that would fall. It is the period in which these cases would all be stretched. This is a familiar picture. The world almost stopped, thinking that we can flatten the graph. Probably we can save a lot of people. And this is another picture just to break the momentum. This was Delhi before the COVID-19 pandemic. This is after pandemic was declared. There are positive stories of pandemic also. We all probably learned to respect the environment, and probably seeing that nature also tries to reclaim itself. And this is a picture. You probably must have heard birds chirping in the morning, more flowers on the plants and things like that. And so I'll just change gears, talk briefly about emergence of zoonotic disease. This coronavirus is a zoonotic disease. What is a zoonotic disease? A disease that can spread from animals to humans or from humans to animals. And why do we get these zoonotic disease? From where why do they emerge? They emerge because of humans. What we've been doing is deforestation, affecting the niche of these animals in the environment. Then illegal, poorly regulated wildlife trade. This is what is expected happened in China. Then climate change is also contributing to this because it affects the niche where the wild animals stay. And then antimicrobial resistance also contributes to emergence of these disease. Then intensified agriculture and livestock production also contributes to this zoonotic disease. One thing more I would like to share with you is, look at these zoonotic viruses, mainly present in two species, rodents and bats. You can say these are the reservoirs. So these are the species that probably contain the maximum number of viruses. But since humans are more on the earth, once these viruses get into humans, you can expect these viruses there will be amplification of these viruses to happen, and there would be global abundance of these viruses also. And that's what has happened during this pandemic. So it originated from Wuhan, now it is all over the world, and you can see what is happening in India now. Another interesting data is bipartite networks between wild and domestic mammalian viruses and zoonotic viruses. You can see all these viruses have interrelationships, and so let's say this is a focus area that I would like to talk to you. There are bats, there are other species. There can be a transfer or a spillover of these viruses from one species to another, because these are all having certain kind of sequence homology. So it may be moving from one organism to another organism, and that's what is leading to then emergence of new disease and new kind of viruses. This is the same concept, but I'm putting it in a different version. This is wildlife. This is domestic landscape, where humans are there, livestock is there, peri-domestic wildlife is also there. Peri-domestic wildlife means what was seen in Wuhan, that they would bring wild animals and then use them for as livestock also. And you can see there can be exchange of viruses between all these categories when we talk of biosphere and domestic landscape. So this is what is the nutshell. I'm talking spillover of the virus from one set of organisms to another, and this spillover can also happen from wild to domestic and from domestic to humans. But interestingly, within the humans, we've seen there is an amplification that has happened, and the virus got multiplied so many times. This is just one illustrative example to say the pathogens could be transferred because of intensified farming or livestock. So people engaged in this, let's say chicken farm, would be doing different kinds of activity where they could get exposed to a virus that could be there in these poultry, and then this would spread to humans. In poultry, there was an advantage. We saw that there were viruses that emerged from poultry farms, and you could do culling of the birds. But this is not possible once this virus is there in the humans, and you can't do anything except for looking for preventing the spread or finding a possible treatment. Talking about pathophysiology of this virus, I try to say that there is one protein that is very, very critical in this virus entry, and called ACE2, angiotensin converting enzyme 2. And this is where it binds to this S1 and S2 proteins. S2 domain, we said, is involved in interaction of this virus with the host cell. S1 protein is also facilitating binding to this protein. What is the ACE angiotensin converting enzymes? These are enzymes that are involved in regulation of blood pressure, I can say in other layman language. They convert this angiotensin 1 into angiotensin 2, which is playing an important role in regulation of electrolyte balance and controlling blood pressure, I can say. So based on the levels of these, we can expect vasoconstriction, vasodilation, and there could be other effects of this also. So virus is using this ACE2 to facilitate its entry. And any cell that contains ACE2 is what is making the entry possible. I said that one thing I said in the beginning was this spike protein is cleaved by a protease, and you can see this ADAM917 is a protease that would act on cleaving this enzyme into these two domains S1 and S2. That is what is responsible for facilitating the entry of this virus into the host cell. Further than this, once the virus enters into the cell, it undergoes proteolysis. The RNA is copied into plus strand, and then you can carry out translation, as well as there could be other host machinery other proteins that would be synthesized, and then we assemble the virus, and then the virus is released. So this is something that is shared by many other viruses, and coronavirus is also using the similar kind of machinery. And you can see these non-structural proteins being synthesized. There could be this 3CL-PO or MPRO that I talked to you in the beginning. All these kind of proteins are being synthesized within the virus. So briefly about this angiotensin converting enzyme. It is a functional receptor for SARS-CoV-2, which is a monocarboxypeptidase. It is a trypsin-like enzyme. It was discovered in the year 2000 and has homology with classic angiotensin converting enzyme. So that means there are variants of angiotensin converting enzymes. There would be some that would be present in the kidney system, some would be present in other tissues. ACE2 is widely expressed in most of the tissues, but if I have to highlight, I will say respiratory tract, intestine, and testicles. And you can understand why it affects. If some of you follow the literature, we always give this corona or corona vaccine to these dogs also, for the reason this coronavirus affects, that coronavirus strain affects intestinal system, causes diarrhea, respiratory tract. These are the two kinds of system through which the virus is able to seek its entry into the host cell. Then I said this ACE2 is involved in conversion of these peptides angiotensin 1 and 2 to their derivatives, which are active molecules that this would get. And angiotensin converting enzyme is also a target for some of the antihypertensive drugs that are in market nowadays. And N-terminal of this angiotensin converting enzyme, I said already, binds to spike protein S1 domain, and binding of this virus particle is endocytosed. And if there is loss of function of this, there is increased level of angiotensin 2, which is then interfering with or altering the blood pressure. Then ACE2 expression is increased by certain drug molecules also, and drugs like statins, which are taken to lower cholesterol levels also. So it was the ACE2 expression which is the critical thing which will facilitate the entry of the virus. There are conditions in which the ACE2 expression would be higher. There would be conditions in which the ACE2 expression would be different, and that would also decide people who get infected and people who do not get infected. Another picture. So this is what I have said, ACE2. This is the ACE2 virus entering, and we know I said this controls blood pressure, and this is how the virus enters into the cell. So if I say the tissues that would be affected by this, you can say it affects cardiac tissues, it affects lungs, it affects kidneys, it also affects brain, and it also causes sepsis. And it involves this, once this virus is infected, it leads to one of the key things is cytokine storm, and that means hyperimmune system activation. That is what is fatal in individuals like this. But I was trying to say that different individuals are affected by this virus differently. We also have seen some people stay asymptomatically, some will not recover, and there is nothing hard and fast that has been said about this virus, but there is some science that is emerging that is indicating that why certain people are affected differently. So we know certain conditions are increasing the risk. For example, age. People with higher age are at a higher risk. Obese people are also at a higher risk with BMI more than 30. People with type 2 diabetes are also at a risk. People with hypertension, you can link it with ACE2 expression. People with cancer, chronic kidney disease, chronic obstructive pulmonary disease, immunocompromised individuals. As you know that immunosuppressants are given to people who are undergoing solid organ transplants. So these are all people that would be having higher risk of COVID-19. Serious heart conditions such as heart failure, coronary artery disease, cardiomyopathies, sickle cell anemia, all are people with all these conditions are again at a higher risk. So varying severity, there is some science emerging indicating that DNA could be the reason, differences in the DNA could be the reason. Something that is most interesting I found was blood group, relationship between blood group and COVID risk. So majority of you are people from science. I don't have to tell you about this, but just to refresh, we have this ABO blood group system, type A blood, type B, type AB, type O. AB means we don't have any plasma antibodies because there are two antigens, anti-A and anti-B, antigen A and antigen B. Type O, no antigens, that means we have both antibodies against A antigen and antibodies against B antigen. The literature is suggesting A blood group is at a higher risk, while O blood group is shown to be protective. And 1,600 patients from Italy and Spain, it was seen that 50% of the patients on ventilator were having A blood group, indicating that this is something, there is a relationship between this. Another study from US at New York Presbyterian Hospital tested almost 1,600 people and found that 33% of the patients who had A blood group types and O negative, O positive were less likely to test positive for coronavirus. So why blood group type might increase or decrease the risk of getting COVID-19 is not known, but some new studies are emerging which are suggesting that anti-A antibodies help to inhibit the virus entry, and it means that, and even O blood group would have this anti-A antibodies, so that means it will help to keep the virus out. And these are kind of preliminary studies. We still need to understand it more, whether there is a true relationship between blood group and COVID risk. Coronavirus and sex hormones. It's been shown, studies emerging showing that baldness may be a risk factor. Men suffer, we all know men suffer more from chronic conditions that are heart disease and diabetes. This could be one reason why you have more COVID-19 in men. And another reason could be men's immune system is not as good as women's at warding off severe viral infections. So we all know this estrogen is protective because it is an antioxidant. It has all good, many good functions that could keep a person protected. But the new study is suggesting that sex hormones might have a direct effect on SARS-CoV ability to enter our cells and establish infection. So I'm going to illustrate this with an example. So data is showing that bald men are more likely to die after they get coronavirus than their hairier counterparts. Male pattern baldness is strongly associated with high levels of testosterone, and along with testosterone, there is another derivative which is called dihydroxytestosterone, which is also in higher amounts. So this is a data which says normal hair person, the risk is 1. Slightly bald person, the risk increases. Moderate, it further increases. Severely bald, you can see the risk increases almost 1.5-fold higher chances of getting serious effects of coronavirus. And this picture we all talked about, this is the ACE2 receptor, which is facilitating the entry of virus into the host cells. And it's been said that this testosterone stimulates this protease, which is TMPRSS2. And we know this TMPRSS2 is, once it is stimulated by this, that means it will cleave this spike protein, and that means it will facilitate the entry of the virus through ACE2 mechanism. This is one of the reasons that has been suggested to be involved in why bald people or people with male pattern baldness are at a higher risk of having fatalities because of COVID-19. Another study that supports this is androgen deprivation therapy is given to patients suffering from prostate cancer, because you have this testosterone pathway targets in prostate cancer also. It has been seen that people on androgen deprivation had a slow, once they were given androgen deprivation, you could see the infection rate was 4 times lower in these individuals, indicating that there is a relationship between androgens and risk of COVID. Another interesting thing, babies are more resilient to coronavirus, which could be something that we may not be able to understand why, because we would see they should be more vulnerable. But interestingly, new facts are emerging that saying few ACE2 receptors in babies. And once there are few ACE2 receptors, you know that there are fewer entry points for the virus into the host cell also. There could be more factors. One of them is HLA types. HLA, human leukocyte antigens. We know there's so much of diversity in these antigens. They are present on human chromosome 6, and there are so many thousands and thousands of types of HLA types that are involved. And we know that they are very, very critical in immune response, because they are involved in antigen presentation. They will bind to peptides coming from viruses, and then this is a virus infected cell, and this is, you can say, HLA type presenting a peptide for host immune response. And you can say HLA type is a sensitive indicator, or you can say alarm system for immune response. And it's been saying that specific alleles, there could be some alleles that could be making the fight against COVID-19 more effective. That means you can expect some HLA types to be, individuals with certain HLA types to be more affected compared to some of these other HLA types. This is a picture of HLA protein bound to a piece of virus. And this is a viral peptide from COVID-19, where you can see HLA protein is bound. So we are saying that certain HLA types will bind more effectively to this virus proteins and then present and have an immune response against these. And you can see this is the antigen presentation by different HLA alleles. You can see some of them would present more variety of peptides, whereas some would have less peptide presentation, and of the oligomers that would be generated from the SARS proteome, indicating that we can expect HLA type to be critical in dictating the host response. And probably this slide is just an error, so we can move on to the next one. Other thing that we know about this is previous immunization. I think when the coronavirus came, we all believed that since Indians are immunized with BCG, we may not be affected by this virus. And to some extent, this is true. We probably have lower mortality from coronavirus than other countries. So you can see the countries which had this BCG program had fewer cases and fewer deaths. And countries hit hardest by the disease are Italy, Netherlands, and US, which do not have a universal vaccination program, and they don't have a TB vaccine program. And maybe I'd like to talk about this slide first. You can see these are the countries that had never received BCG vaccine. You can see the infection and mortality in these countries have been more compared to where the BCG immunization has been given at birth. The graphs have been almost, you can say, flatter. If you split this data into cases per million, this is with BCG, this is without BCG. You can see incidences of cases is very high. Deaths, this is with BCG, this is without BCG. You can see many more. The numbers are very higher. And this is mortality. Mortality is even higher with BCG, without BCG. Going back to BCG, we know BCG is a vaccine against TB, but it does many other things. For example, it also is protective in malaria, sepsis, respiratory infections, leprosy. It also modifies the epigenome that could be helping us against preventing C. albicans infection, staphylococcus Staphylococcus also, and there could be other microbes that could be, because it can generate a cytokine response which could be beneficial. And coming to the last part of my presentation is therapeutics for this. We've been looking for therapeutic drugs for COVID-19, and I think the whole of the world is looking at that. And there can be two possibilities that we can have. We can have antiviral drugs, we can have vaccines. And on both fronts, there have been efforts worldwide to find molecules that could be effective in preventing COVID spread of this virus. And I would like to say that this slide indicates the different targets that we can have to develop therapeutics for COVID. You can have soluble ACE. Soluble ACE means you are blocking the capsid proteins by giving the soluble ACE, which could be the recombinant one, and then virus doesn't enter into the cells. This could be one possibility. You could have drugs that could block this protease, TMP-RSS, or furin-like protease, and that means you're blocking the S1, the spike protein conversion to S1 and S2 domains. Then you can have statins, could also be useful because they can interfere with ACE2 expression. Then you could also have hydroxychloroquine. We don't know too much about hydroxychloroquine. Probably it is inhibiting the proteolysis that is required for once the virus endosome enters into the cell. Then there would be drugs that can inhibit this proteolysis by MPRO. Lopinavir is a drug that can interfere there. There can be drugs that can interfere with replication, and remdesivir is a drug that can interfere with the replication. There are other approaches. There could be molecules that could inhibit the cytokine storm. There are people are trying to work out with gels that can bind to cytokines, prevent cytokine storm, and then help in curing of or preventing the harmful effects or lethalities of this virus. Interestingly, I'd like to share something that came sometime back. There was a paper on hydroxychloroquine which said it is not at all beneficial. Later on, it was discovered that this data was incorrect. Maybe hydroxychloroquine is effective. We still don't know too much about hydroxychloroquine. We need to have more systematic studies on hydroxychloroquine also, but there is a possibility that it might function. So when this kind of situation, anything that even is effective to 30, 40%, 20% is also something that we look forward to. So the more systematic studies required on hydroxychloroquine and other molecules also. Even remdesivir, which is said to be a molecule which is effective, we know that it's not 100% effective. It is only improving survival by 20, 30%. Then there is a race for corona vaccine. We know there could be different approaches that we can use for developing these vaccines. It could be recombinant proteins, viral vectors based vaccines, DNA, RNA vaccines. There could be live attenuated and inactivated viruses. I'll not get into all these, but all these approaches are being tested. You are trying to use nucleic acids also. MicroRNA is one of the approach that Oxford vaccine is talking about. Then we are thinking of viral vectors as vaccines, protein-based vaccines, and then viruses itself, which could be attenuated ones, could also be used as vaccines. And there are some positive stories that we know of vaccines, and you can see the global efforts in developing vaccines, virus-based vaccines. There have been 5+ candidates in trial. Viral vectors, there are almost 25+ candidates in trial. Nucleic acid-based, 20 molecules have been tried. Protein-based, more than 30 molecules in trial, and there are other approaches being tested also. Protein subunit vaccines are also being thought of. And my slide is frozen, so you'll have to bear with me for a minute. And I would say the ultimate, we don't know, since you saw a slide where we said this virus is mutating fast and hello.
Yes, doctor.
Yeah, can you hear me?
Yeah,
you are audible.
You are audible.
I will keep speaking. My slides are frozen. So I said that virus is mutating fast. You have so many different types of this virus strains in India itself. So if the virus has come, we still don't know whether it will give 100% protection also. the ultimate thing would be to have maybe herd immunity. Herd immunity means once a virus is attacks, when it comes back to the individual again, there is a possibility that this will may not be causing the disease again, and the virus becomes weakened, and that's what is the hope. And I would say that probably was my last part of the lecture. I would say we still we still have a long journey to go as far as finding a suitable vaccine is concerned. And I would say, like to quote Darwin here, who said, "It is not the smartest of individuals that survive." I think we need to adapt, to learn and understand about this virus so that we have effective therapeutics available against this virus. And with this, I would like to thank you all, especially AMI and Department of Microbiology, for giving me this opportunity to learn about this virus and share some of my thoughts with you. Thank you.
Okay, thank you, Dr. Sandhir, for the highly informative and nice presentation. The lecture has been well responded on the Facebook, and I'm sure this might have added to the awareness of the audience. And we have certain queries from our participants on the Facebook. I'll ask Dr. Naveen to state a few of these and coordinate the discussion session with the speakers and deliver the vote of thanks thereafter. So
Dr.
over to
Naveen, can you give
Naveen.
me a minute? I can close, probably need to shut down my computer because I can't see anything now.
Yeah,
Or should I
you have to
keep,
state
or should I keep, stay in the audio mode? What do you say?
No, I think you are audible. Your picture is visible. We don't
Okay,
need your
perfectly all right.
Is it
Perfectly
okay
all right.
with you?
Let's go on then.
I have removed your
And
slide.
I welcome Professor Prince also. He has also joined with us. So I think Dr. Rajat, can we have the discussion?
Sure.
So Dr. Rajat, it was a beautiful lecture. It was the best part. I could say that you talked about lots of things. You talked about what this pandemic, how it is spreading, how its structure of this particular virus, then zoonotic diseases, then about the vaccine part, then its pathogenesis, its prophylaxis, its treatment, whatsoever we can give. So you very much justified the title overview, but you are a liar. You said you know the least. I think you knew the maximum after present today in the lecture. So, uh, with you have lots of queries we have. Some of the queries, because now your computer is off, you cannot see the comments. I will take some of the queries. Uh, One is you said that it is particularly reservoirs in the bats and rodents. Is there any specific region you think that why in these two we have the reservoir? Somebody is asking.
Very interesting. Very interesting. This is a thought-provoking question, I would say. I probably don't have an answer for this, but it's a question for which we need to find an answer why it would have so many viruses reservoir in bats as well as in rodents. No clue as of now. No clue as now.
Yeah, I think a good source of research for zoology people.
Yeah.
So they to think about it that why.
Can I just put in Naveen?
Yes, yes,
Dr.
sir.
Prince, I can hear you.
Yeah, क्या हाल है डॉक्टर साहब कैसे हैं?
बढ़िया.
See the bats, they have the cells which have lots of receptors for these viruses. Same way, now it's actually the spike protein in the virus and the receptors on the cells. Those only animals which have the receptors on their cells, they can be infected. And similarly, humans also have developed the receptors. So this spike protein binds. So wherever these animals, those animals which have the receptors for this virus, they really are the host. They bind to this virus strongly, actually.
Yeah, could be one of the reason. I didn't dig the literature, so I probably don't want to comment. But yes,
So
could be possible.
it is believed that compared to SARS-1, SARS-2 binds very strongly to the receptors on the human cells. Actually.
That
That's
is
why
true.
it's so infectious. It is so infectious.
Oh, okay. So thank you, sir. That's a good information. What Professor Prince has told. Sir, you have any other query or any remarks, Professor Prince?
No, he has very elaborately given this lecture. I mean, I'm really very happy that a biochemist has done a very good job which a microbiologist should have done. So, I mean, he has very,
I,
very
I,
elaborately
I was
done this.
scared of you guys. Actually, I was scared of you guys.
No. You have done it very well, sir. Very
Thank
well.
you. Thank you.
Yeah, and before I ask some more questions, one of our student is having some questions. I am doing an experiment, Dr. Soni, today. I have asked her to join the meeting, and let us try that if it works in this lecture. So in the
Yeah,
meanwhile, you please allow Anshika to join. In the
I'll,
meanwhile, I will ask.
I'll allow.
So, Professor Rajat, you said that
Yeah.
uh, these vectors can pass on these viruses to the human beings and all that. So again, maybe not a question of your domain. Somebody is asking that any specific vectors or what type of vectors can be transferring these disease to the human beings and all that.
See, I would say the viruses have always been being exchanged from one species to another one. Some of these would go unnoticed also.
Anshika,
If
join.
they don't cause a disease that is devastating, they don't have spread. So there would be viruses that would be coming. Let's say I'm working in a field where I'm exposed to a new virus from the host or the domestic animal. So there is a possibility that I might fight this virus, I might eliminate this virus. So we'll not know too much about those. The ones that cause pandemic, that is something would come very rare. So viruses have been coming and going from the zoonotic species to, or you can say exchange between animals to humans and humans to animals. But anything that leads to pandemic is where the world efforts are also there. You will not see so much of science done in any other organism as has been efforts have been put in particular in this case. So more is the scare, more is the difficulty, more efforts of worldwide efforts are also there. So there is a possibility that this could be happening all the time.
Okay. So I think Anshika is not able to join us, but I will ask her question. She's asking that it is said that as the number of cases will increase, there will be a plateau in the curve and all that. So how these two things we can say that they are related? That recovery rate will increase as the number of cases will increase. So she wished to know that what is the relation between these two, if I am able to put her question in the right perspective.
So can you keep your mic closer to you because your mic is far, I think.
Yeah, Dr. Rajat, Anshika, our student is not able to join. She is asking that
Hmm.
how the recovery rate is related to the getting plateau to the number of cases. कि अगर अगर नंबर ऑफ केसेस इंक्रीज होंगे क्या प्लेट्यू आएगा कब में न्यू केसेस में
I think some point of
अगर
time we expect some point of time we expect this to happen. The graph has to flatten. There's no doubt about it. The virus will not stay there forever.
okay so
Once
other
the herd immunity develops, we can have a
other
second another peak also. 6 months, you can graph can flatten. We might have another peak coming again, but at the most it can stay for a year, or you can say the virus would be weakened virus then. So it's hard to predict anything. Initially, I was talking to people from Italy when Italy had the highest number of cases. They said India would be in a better position because hot and humid weather is what is bad for the virus. But we've seen that that is not true. So everything about this virus is going to are changing our thoughts, changing our myths. Some days WHO says something different, then they say something different. I think what I am feeling is the vaccine or herd immunity are the best hopes for us. So about predicting the graph, India is a huge country. We can't predict when the graph will flatten or when the plateau would come.
yeah let us hope for the best
We can hope for the
other
best.
more yeah other more technical question she wished to ask is that can the cytokine storm in the severe cases can give birth to more other autoimmune diseases also in the person.
Very interesting. Very, very interesting. See, one thing I would like to say, I cannot answer your question because I don't know what is coming from the field, but they're saying, as in the case of herpes virus, we see the effects of the virus are long term, they can last over years. Even the effects of COVID might last for months and years together. There's a possibility. You might have seen this even in chikungunya and other kind of viruses that we get. The effects last longer than the disease itself. The virus might be gone, the effects stay on longer. So developing this autoimmune reaction, there is a possibility. We will have to have a cross-sectional study which would be long-term, that would tell us what you asked is true or not. At this stage, difficult to answer your question.
Okay, so as you said, lots of things we don't know. And there are lots of questions. I will take just one or two more before we wind up. Rest, we can give your phone number and people can keep on bothering
Sure,
you.
give my email ID or even my phone number. Yeah.
I can give. One of our friend, I don't know that person, Prince Mallory, has asked lots of questions and comments. I will take some of his comments. He said that if we give hydroxychloroquine, it might trap the virus into the cell. So we should avoid it giving to the infected people. It should be only used to the high risk person. What are your comments over it?
See, I am just so about hydroxychloroquine. We had all kinds of stories that are there. They are saying that it can be preventive. You give it to people who are exposed to this virus. It is beneficial. I don't want to comment on anything for which the literature is not clearly. There are no clear evidences that have been given, and no scientific studies that have been clearly done on these kind of things. So whatever we know is kind of sketchy information. So based on sketchy information, I don't want to draw any conclusions. But if you ask me, I would say that maybe hydroxychloroquine works, but looks like scientifically, I would say remdesivir looks like more promising molecule, because we know specific targets, and probably still probably from literature that it can only improve to something like reduce the mortality by 20 to 30%. So that's also something which is a plus. Initially we talked about ventilators, now we don't talk about ventilators because it's been seen when you put people on ventilators, you're increasing their chance, reducing the survival there. And so everything is kind of in flux as far as this virus is concerned. So difficult to say, since the studies have not been done systematically with the large cohorts.
Okay, so Dr. Soni, as you can see in the comment box, lots of questions. Can I take one or two more? I will take one or two more. There are lots of queries regarding this ACE2 inhibitions. How much side effects it can have? So people are just interested.
How much?
How much side effects it can cause if we do the ACE2 inhibition and all that?
See, ACE2 inhibitors are already taken by people. They take it for hypertension. So they're already there. So only thing is, people who are on these ACE inhibitors, it depends on what is the level of ACE2 in those individuals. If the levels of ACE2 are higher, you are at a higher risk. So anything that can suppress ACE2 expression would be beneficial. So you can think of strategies to bring down ACE2 levels. You can give peptides that can block the binding of this S1 subunit to ACE2. So literature is suggesting ACE2 is critical.
I will take the last query before we wind up.
Okay.
So, which is related to myself also. You gave a very interesting information that fat people are more at risk. So, what about the fat people?
Yeah, fat people also, I said in the beginning, it is a risk factor. BMI, I said BMI more than 30. And see, the reason fat people are at a higher risk is because if they have a lung insufficiency, it will be difficult for them to recover because they will have to exert more for breathing. And that's where the challenge is with fat people. So in US also, if you compare the data, obese people were the ones who had more mortality. So fat means BMI. So I would say we can all calculate our BMIs.
So along with Dr. Soni and Prince sir, I have also to be very careful.
So we are more prawns, that means.
Anyway, I have to take excuse from number of our viewers, Sonam, Prince, and Dr. Kanti. There are lots of queries. So we will take up that later on, or we can have some separate section with Professor Prince. So I think he has answered some of the queries which we could take over because time is also limiting. So thank you very much, Professor Rajat. It was a beautiful lecture with overview. Any final comments from Professor Prince regarding this discussion? Any final comments from Professor Prince, please?
Okay, thank you, Naveen. I was a late entrant into this actually, but Dr. Rajat, as a very dear friend and our savior also at many times, and he has taken up our request and he has given a very detailed and nice talk about this very pertinent times about this COVID virus. And he very nicely he says that there are lots of questions, but nobody can answer this because things are changing every day. Even WHO is changing their stance every day. Things are changing. Hydroxychloroquine, the Lancet paper, first it showed that hydroxychloroquine is very effective, and all countries started making hydroxychloroquine, including India. And then the Lancet paper was it was rejected actually. I mean, it was just withdrawn, and then people forgot about hydroxychloroquine. So I mean, things are so different, and people are asking about the plateau and all that in India. The things are very complex. The population, the genetic factors, the recovery rates, they are so difficult. I mean, it's very difficult to answer all the questions. Only some predictions can be made that if we touch the herd immunity to a high level, then more than 40-50% will start touching the maybe start touching the plateau. But before that, it may be difficult unless we get the vaccine, which is more and more vaccine when it is given, more and more immunity will come to the people. But let us say what is in the future, how good the vaccines will be, how nicely they will be working. But at the moment, whatever the information, latest information was there, Dr. Rajat has very well provided it and answered the questions very well. So we are very thankful to him that he has accepted our invitation and given this beautiful talk. Thank you, Dr. Sam. Thanks a lot.
Thank you, Dr. Prince. Thank you for wonderful words. Thank you.
Yeah.
And
So
summing up
before
my talk in very nice words. Thank you.
yeah, before we disperse, I have to propose a little formal vote of thanks. I am first of all thankful to the whole team members of AMI Chandigarh unit, especially all the support and everything given by Professor Prince Sharma, Professor S.K. Soni, and all other members from Punjab University and other institutes for making this lecture series successful. And we are having that about 10th webinar today. And because sustainability of any program is difficult, it is the hard work of the whole team of AMI and chairperson and faculty department of microbiology. So I'm thankful to all for making this series viable. Then no program can be good if there are no viewers. I'm seeing lots of people are following live us on the Facebook, and even after the lecture, people are seeing following our lecture in our YouTube channel and Facebook. I'm thankful to all those who are joining. I'm thankful to the students and all the faculties from other institutes. And in the last, Professor Rajat, I'm thankful to you. Yes, Professor Prince has already said a lot. You took out some time, you gave such a good lecture. And as Dr. Soni has also said in the beginning, he is a scientist, he's an administrator, he is a wonderful human being, and he is an excellent friend. So I'm
Thank you.
thankful to you. I will end this my today's vote of thanks with a couplet which I wrote for Dr. Rajat. वक्त के साथ हर्षा बदल गई, मेरा दोस्त फरिश्ता रहा. Thank you very much.
Thank you, Dr. Mean. Thank you, Dr. Prince, Dr. Soni. Thank you all. Thank you.
Yeah. Thanks a lot.
Thank you.
Okay,